Hello, hello. Good morning, everyone. Today, it is a pleasure to me to introduce the speaker, Dr. Aundy Kumar. He is one of my senior colleagues in the Division of Plant Pathology, Indian Agricultural Research Institute. He did his BSc and MSc from Tamil Nadu Agricultural University, and he obtained his PhD degree from this prestigious institute in the year 1997. Then he joined as scientist in Indian Institute of Spice Research, and after that he moved to IRI as a principal scientist in the year 2010. He did a postdoctoral research work in Wageningen University, Netherlands. He is a very good teacher in our division. He is one of the finest teachers, and you will able to see his teaching ability today. And he is teaching several courses in our division. One of the important courses is molecular host pathogen interaction. And he guided 6 MSc students and 5 PhD students. And presently he is guiding 4 PhD students. He is having research paper in different international journals of repute with high impact factor, and his HI index is 18, and I20 index is 27, and total citation is 1105. With this brief introduction, I welcome Dr. Kumar to deliver a talk on application of this genome editing technology to mitigate the biotic stresses, particularly fungus and bacteria. Thank you, sir.
Thank you, Dr. Anirban, for a nice word about me. Welcome all the students. This lecture is all about genome editing technology for fungal and bacterial disease management. What I'm going to do here is let you know how this plant really interact with the pathogen, or how the pathogens really interact with the plants. This is so important for all of you to make a choice, because we have a technology right now, we can edit anything which is DNA, right? So technology part is well set. Anybody can do this genome editing now. And that is really witnessed in a number of applications which is coming on a daily basis, right? So the focal point of this lecture is I would try to make you understand how pathogen really interact with the plants. In the process, how many plant genes are really affected by the pathogen invasion. So perhaps those genes are so important for genome editing, right? So let's move into the lecture now. The title is Application of the Editing Technology for Enhancing Crop Resistance Against Fungal and Bacterial Diseases in Crop Plants. And I just would like to have a quick recap of what we have done in the last 10 days. So maybe in few slides, the quick recap of the definitions and various GET tools. That would be followed by my own pet subject, the threatening diseases of crop plants, what we need to really focus now, and why genome editing tool is important or vital for crop protection, maybe the agriculture version 2. And what is a cellular, genetic, and molecular basis of susceptibility and resistance? These two are the two contrasting terminologies in plant pathology, right? So you should know what is the basis of susceptibility, which eventually culminate in a phenotype, what is called as symptoms. And you also should know what is the basis of resistance, which is the most accepted or most desired trait in agriculture, because if the crop is resistant to pathogens, then definitely you can secure the so-called food security. And the choices of genomic loci or genes for editing that may lead to the desired phenotype. So in this case, it is no disease situation is the one we really want by doing all editing technologies. And of course, number of success stories. I'm really spellbound after seeing all those success stories which is coming on a daily basis, right? I'll take you to the tour of a series of slides where people have confirmed using some kind of editing in some plant genes. They have come out with a kind of product which is ready in terms of achievements. So let's take a tour of those achievements, and of course let's make a kind of a comparison between the genome editing technology and also the conventional technology, where we really stand in terms of efforts and also the end product. So this is a flow of this lecture. Perhaps it might take an hour or so. Let us deal one by one. Just to recall once again, I think all of you are quite familiar now. What is genome editing? This is a very apt definition. I could say it's a very broad-based definition, a collection of advanced molecular biological techniques. We can put it as a genomic techniques also, to facilitate a precise, efficient, and targeted modification of a genomic loci. So the genomic loci could be anything. It could be a kind of a coding sequence, or it could be a kind of promoters, or it can be anything which you would like to meddle with, right? And what's happening in those loci? That's in the second page. That is insertions, maybe deletions, or point mutations. You think of a kind of a type of mutation, you can really inflict those things in the genome, right? So this is, I just borrowed from this particular author, Hala et al. 2018. And of course, you might have been given a lot of input by Dr. Nirban and co. The genesis of the whole technology, what we have right now. I think perhaps we cannot ignore the zinc finger nucleotides that was to start with. And it is a kind of early years of genome editing. So we have a zinc finger nucleus that pair bound to DNA. I think they're all exposed to all these things, Anirban, I'm sure, right? Yeah. And this is something like, the DNA binding domain at the N-terminus. There is a zinc finger protein, and there is a C-terminal. There is a kind of enzyme, what is called as FOC1. The FOC1 stands for bacterial enzyme, that is Flavobacterium echinocoides. So this is the ring nucleus is borrowed from this particular prokaryote. So what's happening here is the targeted sequence of zinc finger nucleus pair are typically 18 to 36 base pair in length, excluding the spaces, right? So this is the cartoon of how it really looks like. And we have the parallelly, another one came to literature, what is called as talents. And that is a transcription activator like effector nucleases. And again, these two are almost look alike. So you have a kind of FOC1 enzyme on the C-terminal, and you have on N-terminal, you have tail. So this is a kind of configuration of this particular protein. Again, it does the same thing like the zinc finger protein. And I also come across another kind of nucleases, what is called as meganucleases. And again, there is a great deal of literatures available. I appeal to all the students to keep abreast of this particular terminology, meganucleases. And this is a very unique enzymes. It has got a kind of very long recognition site, and there are two different kinds. One is called intron endonuclease and the intine endonucleases. Please enlarge your knowledge on this particular subject. And the focal point of the training program, and also my own focal point here, is the CRISPRs, that is clustered, regularly interspaced palindromic repeats, right? And this is creating waves in the subject biology now, in any field of biology. So this is the cartoon, how it looks like. And the key feature is the CRISPRs were originally discovered in the late 80s. Ishino et al., I think Dr. Nirban might have given the historical account of this particular discovery, and originally discovered in Streptococcus pyrogenes, and presently it is making a lot of impact in biology, in particular agriculture and human medicine. So this is a nutshell about the CRISPR-Cas nuclease. It's bound to a synthetic guide RNA, sgRNA, complementary to 200 base pair target in the genome. It can be anything. It can be a coding sequence, or it can be any non-coding or promoter. The base pairing between the DNA sequence and sgRNA after protospacer adjacent motif, what is called a PAM motif, recognition allows the DNA cleavage at the target site by Cas9 nucleus. So I think you must have already understood The PAMs are so important, they only attract the Cas9 protein, and that takes the RNA to the complementary sequences in the genomic region, where it inflicts a kind of what do you call restriction. It may be a kind of double cut or single cut, whatever. All those things you might have quite familiar now. So the question here is, what this Cas9 is really capable of. That is very, very important for you to understand. It can create any kind of mutations, which I already told you. So the Cas9 or the modified versions of Cas9, what I want to make a point here is already the modifications have come in Cas9 protein. They are all tailor-made for specific applications in crop sciences. The modified nuclease variant can inflict changes in the arrangement of nucleotide bases in the genes or genomic loci for that matter, right? So what kind of changes? It can inflict indels. The single guide RNA Cas9 complex can induce a double-stranded break, the famous DSBs, on the target locus. So in the process, what happens? DSB is primarily, any host, the DNA is attacked, they have an inherent mechanism to repair it, right? One such inherent mechanism is what is called as it's a very nice acronym, that is NHEJ, that is non-homologous enjoining method. This is one of the most important, the inherent mechanism in every biological system to repair the damaged DNA. So this is exploited here to inflict or to trigger the mutations in those genomic loci, right? And which can lead to a kind of, it can lead to a frame shift. It can, if the frame shift is there, definitely is going to alter the expression, the protein composition of the one which is translated out of that particular loci. So this is a simple principle behind this. And it can create even deletions. For example, if the Cas9 is going to cut both the strands, and the entire strand is removed, this is what is called as excision of the target fragment. That is also quite a reality under this Cas9 protein. And it can create insertions. You can make the whole system to insert something in the genome. So that is the power of this particular technology. What is called as insertion via non-homologous enjoining. Any NHEJ mediated repair of the double-stranded break can be exploited to insert a donor fragment. If you have the donor fragment designed for your own applications, you can supply that, and that's going to get integrated into the loci of your interest. So this is another option. And insertion via HDR is another option. So if you have the homologous sequences, which is already tailor-made in your target, in your what you call donor, you can even inflict what is called as HDR, that is homology-directed repair. So this is another reality. So, and the variants I was talking about, I stumbled upon two variants. One is the DCAS, that is deactivated Cas, or sometimes it is also called as dead Cas. That means the enzyme lost its ability to cleave the nucleic acid. And we have another one, what is called as RCAS, that is RNA Cas. So these two are the new additions, or new variants, that is going to create kind of applications in biological sciences. So what it can capable of doing, one is transcriptional repression. For example, a dead Cas guide RNA complex can be targeted to a promoter region of the gene. In the process, you can repress the transcription. This is another option. And yet another option is a silencing via RCAS, because a new variant, what is called as RNA binding variant of Cas9, what is called as RCAS9, it can bind to a specific sequence of the messenger RNAs, leading to a translational silencing. What I'm trying to tell here is the silencing can be at a transcriptional level, it can be at even translational level. All of them are going to assist you in editing the genome one way or other, okay? And two other applications of this particular Cas9 protein, perhaps it is again going to make a big impact in biological science. One is the base editing. So base editing is something like, a modified Cas9. For example, Cas9 is cytidine deaminase fusion protein can be targeted to a specific region leading to deamination of cytosine. So if the cytosine is deaminated, what happens is the daughter nucleic acid, what is going to be biosynthesized, will have AT. So that is a modification what you are going to do. So this is another application. And yet another one is epigenetic editing is also a reality using Cas9 protein. So here the dCas, the one what I already told, the transcriptional repression is possible with the help of dCas9. The dCas9 can be fused with additional protein domains to allow for epigenetic editing. So that is, it is done through the two mechanisms. One is the cytidine demethylase, that is 1011 translocation deoxygenase 1, and the histogen acetyl transferase fusion protein can be targeted to very specific sites to enhance gene expression. This is very interesting. So if you want to enhance even gene expression, that's possible using this technology. And the opposite of that, for example, the transcriptional repression is also possible using another Cas variant, the Cas9 with the cytidine methylase, that is the DNA methylase transferase 1 or histone methylase that leads to transcriptional repression. So students, what I just highlighted in few slides is the different options what is available right now for us to edit the genome, right? So with that background, let us move on to the topic for today, that is why genome editing technology is needed for crop protection. So I would definitely make some justifications for doing this genome editing in crop plants for crop protection. So these are all some of the, what do you call, everyone knows about this. Plants are attacked by too many pathogens, right? Too much of abiotic stress. They are always reducing the genetic potential of the crop plants to yield more and more or to give the best quality product for us. So pathogens can cause substantial losses. They threaten the food security. And of course, we know pretty well that the existing technologies are very insufficient to sustain the agriculture production. So all of you must be knowing the two best technology in crop protection, especially in plant disease management, is agrochemicals and followed by host resistance. These two are thoroughly exploited to sustain agriculture production all these days, right? But the question here is, can we really depend on these two technologies forever? Right, here comes the question of their either the durability or the safety issues associated with the chemicals. So that the world is quite aware of all those things. The host resistance is, remember, is not durable, right? Although we claim that we have the best of the best technology to control plant diseases, this is only a short-lived, right? There is nothing like a durable host resistance. So somewhere in some part of the world, some new variant emerges, and that makes the so-called resistant variety, what they call useless. That's a reality. We have a number of examples. I'll just show you in the next slide. And chemicals, not to talk much about it. For example, one recent incident is any chemical residues on your finished product which is meant for export, now it is under scanners now, right? So you should not have any residues on the produce if you want to really market your product. So the best technologies what we are boasting of in agriculture, especially for crop disease management, the host resistance is not durable, and chemical-based disease management is always under scanner, and somewhere people are going to oppose it, right? So therefore, I would say, in my opinion, you need to have innovative crop protection strategies to sustain the agriculture production by reducing the crop losses by the plant pathogens. So again, to justify why these novel methods are needed to protect economically important crops, right? This is very important. So the recent incidents, I would take two examples. One is in North Africa, a crop like banana, there was no record of xanthomonas, right? So suddenly there is an outbreak of a new kind of disease in Tanzania and other places, and that disease, it is caused by a completely different pathogen, what is called as xanthomonas. Till that time, there was no record of a bona fide xanthomonas species affecting banana. So this is what is called as the pathogen somehow found its way into banana crop and started affecting it. It became a serious problem, right? What is called as XPW, that is Xanthomonas bacterial wilt, right? Few years ago, an Asian country, it created a kind of waves in agriculture fraternity. There is an outbreak of wheat blast in Bangladesh, right? And there was no record of a serious crop loss in wheat by a blast disease. So until we experience it in our neighborhood, that is in Bangladesh, what is called as the wheat blast outbreak in Asia. So these two incidents it gives us enough of evidence to say that the crops are not really safe. It is only a time that is going to decide whether the crop is going to be free from the pathogens or not. So what the statement here is, the pathogen, they continuously evolve and adapt. That's the reality, to overcome the host immunity. The so-called the resistant plant becoming susceptible across a completely different species. For example, the pathogen is known in rice and it is recorded in wheat, right? So what is the link between these two? I would say that it's a host jump, right? So the pathogen can emerge on any crop, right? And another example, I think pathology students, if anybody is sitting here, they would understand this. There is a strain of wheat rust fungi, what is called as UG99. It created so much of havoc in, again, some part of Africa. And these two examples are enough to highlight pathogens always evolve and they overcome host immunity sooner or later, right? So that means the host resistance cannot be dependent forever to mitigate crop losses, right? And agrochemicals are not accepted because they are not only the sense they are not environment friendly. They may pose risk for exports. And the classical example is the tricyclosol in rice. It is creating so much a problem for our farmers, right? The elevation of MRL 100-fold in European countries and Middle East countries is a serious issue now, because we know that tricyclosol is an integral part of rice farming, especially in the basmati growing region, because where the blast is a regular occurrence, we need tricyclosol to control rice blast, right? And the residues, what is left on those produce, is a serious problem. Now the exports are being rejected by the importing countries. So that means these two technology, what we have, it cannot be relied upon for long. So this is just to tell you about the different kind of diseases, what is really affecting crop plants. This is a kind of, just to tell you what kind of symptoms the disease, the pathogen can cause. So those who are not too familiar with the terminologies here, we have different type of pathogens that it reflects their lifestyle. For example, we have necrotrophs, we have hemibiotrophs, and we have biotrophs, right? And what I want to say here is the different kind of lifestyle adopted by these different pathogens can really create a serious yield loss in several crops, right? The classical example is Rhizoctonia solani in rice, and we have Magnaporthia and Phytophthora in rice and potatoes, and have biotrophs. We have three major diseases in the world. One is the Puccinia rust, and we have Erysipellae powdery mildews, Plumeria powdery mildews, and we have downy mildews, and so on, so on. So these pathogens, they are too diverse and two different kind of lifestyles. They can really create a serious yield loss. The next few slides, I'll just tell you some of the top diseases which the world is really focusing on now. And to start with the fungal disease. So among all the pathogens, the fungal diseases are they are the most dominant one, because there are 8,000 fungal species. They are capable of causing plant diseases, right? That's too many numbers, right? So we have, starting from Puccinia, we have Fusarium, Septoria, Plumeria. Again, a wilt caused by Fusarium oxyspora, Magnaporthia oryzae in number of cereals, including wheat, and we have Botrytis cinerea. This information, I could just borrow it from one of the flagship journal in plant pathology, that is Molecular Plant Pathology. They have come out with the top 10 diseases in the world, the fungal diseases. I think all of them are highlighted here, right? And come to another group of pathogens. They are equally important. In fact, the subject plant pathology evolved from one of these pathogens, that is Phytophthora infestans. It's a serious problem in potatoes. We have Phytophthora capsicum in vegetables, and we have Plasmapara viticola in the case of grapevine, and we have Phytophthora parasitica in a number of orchard crops, in particular citrus, and we have Albugo candida in cruciferous plants. All of them are very serious problems, and they are capable of reducing the potential of these crops. And again, I just borrowed it from Molecular Plant Pathology journal. And come to the bacterial diseases. They are the small numbers, but they can really create an explosive epidemics in number of crops, right? And the threatening bacterial diseases of crop plants, they can cause a variety of symptoms. For example, spots and blights, cankers, wilts, and rots. And the top 10 bacterial pathogen which we need to really focus, or the research effort should go on. That's listed here. Pseudomonas syringae, Ralstonia solanacearum, they cause bacterial wilt in solanaceous vegetables and ginger. We have agrobacterial tumefacients in stone fruits. We have Xanthomonas oryzae, Pathovara oryzae in rice. Xanthomonas campestis, number of pathovars. And Xanthomonas actinopodis, several pathovars. And in our country, we have Xanthomonas actinopodis, pathovar punicae in pomegranate bacterial blight. It's the number 1 disease. And we have Erwinia amylovora. Of course, we don't have this problem in our country, but again, it's internationally well recognized plant pathogen. Xylella fastidiosa, Dickia, Pectobacterium, and so on and so forth. So what I'm trying to highlight here is, although we have 200 bacterial species are known to cause plant diseases, among the 200, the top 10, what was highlighted, that's here. So they can attack both above ground and below ground portion, and they can really create devastation. So those slides, just to impress upon you the seriousness at which these pathogens damages the crop, right? So I make it a point here for bacterial disease management, the only option which is time-tested and proven is host resistance, right? And again, it comes with the risk of losing the resistance sooner or later, because you know the bacteria, they can short generation time, they can really come out with the huge numbers in the infection foci, and few of them are already a mutant that can really overcome the resistance, right? The pathogenic variability is so much in bacterial pathogens, and any resistance what you bring, it is very, very short-lived, and it is going to, the disease is going to come back to you again, right? With all those background, so let us take the class forward, and there is a strong justification because we have too many problems from the pathogen side. The technologies are, although rated as very high-end technologies, but they have their own deficiencies. So we need new kind of approaches in the next agricultural scenario, right? So, but before you venture into such an effort, in my opinion, what is more needed here is you should really understand what is the basis of the disease in the first place, right? And what is the basis of resistance? That is also you need to know. And what is the mechanisms really operating in either side, right? Definitely there is an interaction. There are two entities are involved here. One is the pathogen, another one is the host. When they interact, something is happening on either side. And definitely it is, if something is happening in the world, definitely it is something related to the gene and the gene products, right? So before you decide to apply the GET for disease control or crop protection, you must understand the cellular, genetic, and molecular basis of the so-called disease and the no disease. The disease is what is called susceptibility, and the no disease is nothing but the resistance, right? In the next few slides, it is typically host pathogen interaction related subject. You pay attention to the slides. Let's come to the first point. How this plant is recognizing a microbe. That's very, very important, right? If you take any plant pot, foliar, you take rhizosphere, you just plate it in a simple nutrient-rich media, you can easily count 10 to 7 to 10 to 8 bacterial species per gram. Oh, that's too much number, right? Right. And on foliar, you would easily score 10 to 6 to 10 to 7 bacteria. And I'm not talking about other microflora what is associated in those niches, right? Just one microbial entity, that is bacteria, is living in 100 millions in the rhizosphere, maybe around 1 million to 10 millions on phyllosphere. The question is, are they inert on the plant pot? Or is there any communication which is happening between these two? That's very, very important, right? So the understanding what we have based on the information what is available in the literature, maybe it accumulated over the last 20, 25 years, the plant recognizes the pathogens, or I'd put it like this, the plant recognizes the microorganisms by a kind of factor what is called as receptors, and those receptors are popularly called as PRRs. PRRs, it is an acronym for a pattern recognition receptors. So what I'm trying to make a point here is plants, they evolved this receptor, right, exclusively to recognize what is coming onto that sphere, right? The PRRs are highly conserved proteins in plants. That means it is something which is so intimately associated with the plant life, right? And there's a great degree of conservation of these PRRs in plants, right? If you take a family-wise, a specific PRR is there in all members of that particular family, right? We'll take some examples in the future slides. What's happening here? When the microbe come to the plant site, the PRRs are the first one to really interact with the microbes, right? So what's happening here? The PRRs interact with the microbes. The question here is, what part of the microbe is interacting with the PRR? That's very, very important. So those part is now called as PAMs, right? It is pathogen-associated molecular patterns. That means much similar to the receptors what is present on the plant or evolved by the plant, these microbes also evolved something like PAMs in their life, right? These two, I would say, the factors are extremely conserved in the respective organisms. The conservation of PRR is there in the plant. It is much similar to the conservation of PAMs in microorganisms or pathogens. So what's happening here? So it's a picture. These PRRs are anchored in the cell membrane, right? You can imagine something like this. So what happens? They are just anchored on the cell membrane. They do not do anything unless and until the plant encounters a microorganism, right? When they encounter a microorganism, so what's happening here is the bacteria has come to the immediate vicinity of the plant and it started negotiating with the plant. So what's happening? Imagine this is a pathogen, this is a PRR. So the moment it comes near to the plant, they interact. What I mean, there is a physical interaction between these two components, right? Literally, there is a physical interaction because the proteins are configured by the plant in such a way that PRRs and PAMs can undergo kind of something like a lock and key configuration. So this is so important. So what's happening? Once they are interacting with each other, the plant open up a completely different kind of metabolism in the host, right? Otherwise, it is silent. So that is what is called as the defense signaling pathways. This defense signaling pathway is popularly called as a PAM-triggered immunity or simply called as a PTI. Right? I hope you understand this. This is the first step in plant microbe interaction, right? Remember, this PTI is good enough for the plant to stop most of the microorganisms, right? They will not cross this boundary, provided the microbe happen to be a pathogen, right? That means PTI is a powerful inherent ability of the plant to keep the microorganism away from its endosphere, right? It works all the time because I already told you have 100 million bacteria which is living in the rhizosphere. Not all of them are entering into the plant, right? We have 10 or a million or 10 million bacteria which is living on the foliar. We have only 1 or 2 xanthomonas which is causing disease in rice, right? That's an extreme minority which is capable of causing disease. The rest all kept away from the plant sphere, or rather they are kept away from exploiting the plant metabolism for their own life cycle completion, right? Try to understand this. PTA is good enough to stop most of the microorganisms, right? But the question here is, what is a PRR? Are they characterized one? What they are chemically composed of? The literature, what it says, we have 4 different types of receptors. Of course, different classifications are there. You will read from literatures. And in my perception, these 4 are based on their chemical nature. We can classify these PRRs into 4 different categories. Number 1 in the chart is RLKs, that is receptor-like kinases, and receptor-like proteins, right? And 3rd and 4th is yet to be characterized, but the reports are already started coming in literature. One is called intracellular soluble receptor, another one is extracellular soluble receptors. These 4 are the major classes of receptors. They are anchored in the cytoplasmic membrane of the plant, and they negotiate with the microorganisms, right? And this is how it looks like, right? Receptor like a kinase. We have a kind of LRR domain which is going to perceive the microorganism or the PAMs of microorganism, and we have a transmembrane domain which gives anchorage for this molecule or the compound on the membrane, and we have there is something like a kinase domain which is the cytoplasmic side of the whole setup, right? This is receptor like kinase. And this is a receptor like protein. The difference is there is no kinase domain in RLK. If there is no kinase domain, the same protein is what is called as RLPs, receptor like proteins. And is there any known RLKs in the literature or in plants? Yes, we have number of RLKs which is already well documented, and we know their function as well, right? This is the list. We have FLS2, that is flagellin sensitive 2, is a classical PRR, and it interact with a pathogen molecule, what is called as a flagellin. And it's not the entire flagellin what is going to interact with this FLS2, it is only a 22 amino acid of this flagellin is going to interact with FLS2. That is called FLAG22, right? So we have flagellin 2 is a classical RLK, followed by EFR, that is elongation factor receptor, NRF1 and NRF2, that is a node factor receptors. We have XA21, XA26, is well known. We are using this in agriculture for mitigation of rice bacterial blight. And PID2, that is resistant rice blast, barley RPG2, tomato SR160, and the PEP R1. These are all well characterized. We know the function of these receptors. They are all classified as RLKs. Let's see some of the RLPs. The classical one is the one which is going to give resistance against rice blast, that is CBIB, chitin elicitor binding protein, right? This protein is anchored in the rice cytoplasmic membrane, and that is going to interact with the pathogen associated molecular pattern. In this particular case, it is chitin, because we know that most of the fungal pathogen, they have chitin in their cell wall, right? Chitin which is completely absent in the plant, right? So the plant can interact with the chitin which is not native to the plant. So this is a well characterized one. EAX1, that is ethylene inducing xylanase in tomatoes, and we have CF2, 4 and 9, the classical one, that is the Cladosporium fulvum proteins in tomatoes. They are all classified as RLPs. Let's move on to the pairs, right? I was talking about the receptors, and these receptors where they are going to interact on the pathogen side. So the well-known plant receptors participating in the host pathogen interaction, the PAMs with the known receptors, I always tell this as 5 examples. One is a FLAG22 is interacting with FLS2, is documented in number of hosts, right? And we have ELF18, ELF26, that is elongation factor 18 and elongation factor 28. These two are receptors. These two are what they call PAMs. They interact with a receptor what is called as EFR, that is elongation factor receptor, that is documented in Brassica family, in particular Arabidopsis. And we have another one, Albertsheim's heptaglucoside. That is a receptor that is interacting with a pathogen molecule that is in Phytophthora megasperma in soybean system, that is extracellular glucanase. And we have another one, ethylene inducing xylanase, that is EIX2 in tomato. It is interacting, the ethylene inducing xylanase is a kind of a receptor that is interacting with the, what do you call, it is a PAM, it is interacting with EIX2, that is a receptor in tomato system, and the chitin that is interacting with the CBIB. So these are well characterized one, and they participate in host pathogen interaction, and the outcome of this interaction in the first step is always PAM triggered immunity, right? So the interaction between PAM and receptor leads to PAM triggered immunity or PTI, and this is also called as horizontal resistance, right? Why it is called horizontal resistance? Let us not forget, it is not FLS2, FLAG22 alone is present in the pathogen side. You have so many other PAMs also. So those PAMs also can theoretically can interact with another receptor in the same plant. So it is always polygenic. So what I'm trying to say here is there are many PRRs, which is present in the plant, they can technically interact with the many PAMs in a particular pathogen, right, or microorganisms. So for that reason, we always consider this PTI as a polygenic and also called as horizontal resistance. And this is a point, the take-home message is the interaction between PAMs and PRR, which leads to PAM-triggered immunity or PTI or horizontal resistance, right? But if that is the end of the story, then there should not be any plant disease at all, right? But the diseases are coming all the time. So that means something else is really operating beyond this PTI, right? So the point here is the PTI is very, very short-lived, or rather it is going to be what they call nullified by the microbe, which happen to be a pathogen, right? So the pathogen can overcome this basal immunity because pathogen also would like to evolve, would like to complete its life cycle, would like to enlarge its population in a system. So they need resources, and the resources are from the plant side. That means they evolved a mechanism to make this PTI null and void. That's what's going to happen, and that is possible by the pathogen by a completely different mechanisms, right? Let's see what is that. The successful pathogen which defeat the PTI and cause infection in the plant, right? And this is done with the help of a powerful pathogen secreted protein. And those proteins are often called as effectors. In other terms, a microbe which is capable of secreting effectors are all called as pathogens, right? And the effectors are coded by a specific genetic loci in pathogens, and they are popularly called as virulence genes. If you read literature, they were originally termed as virulence genes, right? Now we know that they are the most potential virulence factor of most of the pathogens. And this effectors, what is its function? These effectors would try to stop this PTI from happening in the host. It has to do that. Then only it can make further inroads into the host system to exploit the host metabolism, right? So effector can interfere with the PTI signaling and make the plant susceptible, right? And effector target plant cellular hubs and genomic regions or plant proteins. The question here is, what is the effector function in the host? What it does, it targets some plant cellular hubs, number one, and some genomic regions in the plant genome or some plant biosynthesized proteins. So what's happening here? The selective plant targeting, which leads to biosynthesis of or accumulation of factors, which is needed by the pathogen to undergo the so-called developmental transition from one stage to another stage in the case of fungal pathogens, or to enlarge its population size in the case of bacterial pathogens. So they need energy. This energy is obtained from those host factors, and the host factors are specifically targeted by the effectors, right? So this is the most important point. The whole step right from effector delivery into the plast or effector delivery into the cytoplasm till they reach the cellular hubs or the plant proteins to convert what is available there for something which is palatable for the pathogen. The entire step is popularly called as the pathogenesis events or pathogenesis pathway. The culmination of this pathogenesis pathway is what is called as the pathogen colonization, and visibly this is also called as a life cycle completion of a pathogen. And how this is possible? When the spore lands on the foliar part, it is a single spore. In the case of rice blast, after 10 days, a lesion is going to release 30,000 conidia. So how is it possible in a matter of 10 days, a single conidia is going to release 30,000 conidia? So we need resources, we need energy, space, everything is provided by the plant. And this is possible with the help of the so-called powerful pathogen factors. They are called as effectors. It is the handiwork of effectors that make the plant associated factors into something which is palatable by the pathogen to undergo a developmental transition from one stage to another stage. In the case of fungal pathogen, it is the transition is from a vegetative phase to a reproductive phase, in particular the so-called asexual reproductive phase, right? A single conidia is going to produce 30,000 clonally produced conidia. That is a real threat in agriculture, because the 30,000 conidia can create theoretically 30,000 independent infections in the field, right? That is how this pathogen they evolve and they increase, enlarge its population in the ecosystems, right? And what happens? In the process, the plant react. The plant react. So it is not going to be silent because it has to react. And this reaction is perceived by all of us in the form of symptoms, right? So plant react to the cellular events, which is happening much against its wish, but it cannot stop it because effectors are having a free ride into the system, and it is going to convert what is available in the plant pot to something which is palatable by this pathogen, and the colonization is ensured there, the reproductive structures are formed, and plant is reacting to that in the form of symptoms. And remember, the whole event is, end of the day, you start seeing the symptoms, or you start seeing the pathogen directly on the plant pot in the case of rust and powdery mildews. And this whole phenomenon is called as effector-triggered susceptibility, right? This is the second stage in the plant pathogen interaction, right? In plant microbe interaction, the first stage is I would say the first and last stage there. But the microbe happened to be a pathogen. The second stage is triggered, and that leads to a phenomenon, what is called as effector-triggered susceptibility, right? This effector-triggered susceptibility has got very unique phenotype. That phenotype is what is called as disease symptoms. It varies from pathogen to pathogen. A wilt pathogen causes wilt, right? Foliar pathogens, leaf spot pathogens, they cause leaf spots. Some causes blight, right? It varies from pathogen to pathogen and system to system, right? So one important point here is effectors are always expressed when the pathogen is already there in the host system, right? So rarely the effectors are expressed in the pathogen side, right? So the point here is effectors act outside the pathogen cell. What I mean to say here is, as long as the pathogen is not interacting with the plant, effector genes are not going to be expressed, right? They will express only when they already in contact with the plant system, the PTA is already triggered, the next stage they are expressed. Either they are in the apoplast, or they are released into the cytoplasm, right? This effector-triggered susceptibility is the one which is, we know that, I already communicated to you, that is the one what is perceived as a disease symptoms or signs. But this effector-triggered susceptibility can be stopped. How it can be stopped? It can be stopped very nicely, but we have been doing it for many, many years. This is stopped just by entering into a program what is called as breeding for disease resistance. What you do, you bring another factor into the host system which can stop this effector from entering into effector-triggered susceptibility, right? So effector-triggered susceptibility can be stopped by deliberate introduction of functional R proteins encoded by R genes. This is one of the most successful research activities worldwide people have done and come out with a thumping success in many crops, right? All our breeding program for resistance, breeding for resistance program is based on this principle. What you do, you bring a resistant gene into the cultivar, which is going to stop the effector from entering into effector-triggered susceptibility, or which is going to stop these ETS from happening in the host system, right? Let us see how this is possible, right? The plant breeders have developed resistant cultivars by introgression of R genes. So what is this R genes after all? These R genes are, in my opinion, they are activated R proteins, perceived and physically interact with effectors. I was talking about the PRR, physically interact with PAMs. Much similar to the story here. The effectors are physically interact with the so-called R proteins. For the benefit of the students, you must understand R proteins are nothing but intracellular receptors, right? The most of the intracellular receptors or cytoplasmic receptors, what we call now, they are all the product of these R genes, and they interact physically. When this R protein physically interact with effectors, what is going to happen here? The plant disease resistant gene encode proteins are intracellular receptors that serve to sense the invasion of the pathogens. So the point here is the function of PRR, the function of R genes are technically same, right? Well, PRRs are the general perception of all microbes, what is coming to the plant sphere. But the R proteins are a specific receptors. They can interact with those pathogens which entered into the plant system. You got it? So that is the most important point here. So they are very specific receptors. They are capable of interacting with the pathogen encoded proteins. Those proteins are called effector proteins, right? This is happening either in the apoplastic side or it's happening in the cytoplasmic side. So what's happening here? The intracellular receptors that serve to sense the invasion of pathogen and subsequently trigger a series of downstream immune responses. I was talking about the same thing in PTI. When PRR interact with the PAMs, the plant open up a completely different signaling pathway in the plant system to stop the microbe from entering into the plant site, right? Same thing happens here. So when these R proteins are expressed, a functionally viable R protein, they have a protein-protein interaction with the effectors. When they interact, what's happening in the plant side is the plant will not allow the effectors to gain access to those cellular hubs which otherwise would have supported the pathogen to undergo life cycle completion. The effectors will not allow that. So rather, it open up another pathway. That pathway is what is called as a defense signaling pathway. This defense signaling pathway is quite strong defense signaling pathway. This pathway has got several biochemical and phenotypic phenotype. For example, ROS production, salicylic acid production, HR. We have a PR proteins and overall what is called as SAR, that is systemic acquired resistance, to keep the rest of the cells or rest of the tissues free from pathogen. This is a cascade of event which is operating in the plant side. And who opened up this? It is the interaction between R protein and the effector protein is opening this particular pathway to keep the pathogen from gaining access to those cellular hubs which otherwise would have contributed for effector triggered susceptibility, right? So the intracellular receptors are two major classes right now, very popular. One is TIR NBS LRR, that stands for toll interleukin, nucleotide binding site leucine rich repeat, and coiled coil, nucleotide binding site leucine rich repeat. These two are well characterized one. Let's take some examples. The TIR class is well characterized in the case of linseed, that is Melamsora lineae. It is having a specific interaction with an effector. Remember, when I talk about this intracellular receptor or cytoplasmic receptor, what is called as R proteins, they have a very specific effector molecule in a very specific pathogens, right? It's not general at all. It's very, very specific. For example, L5, L6, and L7 are cytoplasmic resistant proteins or intracellular receptors. They have very specific binding ability on an effector which is produced by Melamsora lineae, that is AVR L5, 6, and 7, right? A classical example of TIR. And we have another example, cytoplasmic resistant protein M, which mediate the recognition of Melamsora lineae effector protein AVR-M. Let's take some example for CNBS LRR. The classical example is rice. What is AVR-PITA? AVR-PITA is an effector protein that interact with the effector receptor in the rice. That receptor is rice LZCCNB LRR. So these are all excuse me. These are all the well documented one. And tomato I2 is a receptor which mediate the recognition of an effector coded by Fusarium oxysporum in tomato, that is SIX3. These pairs are well characterized, and the effector and receptor pairs, when they interact, remember, the host is going to open up a defense signaling pathway, right? And that defense signaling pathway is called as effector triggered immunity. So here comes the so-called two diagonally opposite phenotypes and triggered by the same effector, right? The effector in the absence of receptor can trigger effector triggered susceptibility, but in the presence of a receptor, a completely opposite phenotype in the plant, what is called as effector triggered immunity. So these two are the one I was talking about, the molecular basis of susceptibility and the molecular basis of resistance. So what's happening here? The interaction between the effector and a functional R protein, also called a cytoplasmic receptor, or intracellular receptor. By and large, they belong to a class of protein, NBS, LRR. When they interact with the effector, the outcome of this interaction is immunity in the host. And that immunity is ETI, very popularly called as vertical resistance. Remember, vertical resistance is the one which is thoroughly exploited in agriculture to bring out the varieties which is resistant to number of plant pathogens. It's so successful in wheat breeding programs, so successful in rice breeding programs, and many other crop which is so important for food security, right? This is what is called as vertical resistance. Just to sum up the story, what we have been discussing in the last few slides, to start with the PTI, we entered into ETS, and finally, with the help of breeders, we triggered ETI. Just to sum up, I'll take you to 1940s, 1945 to 1950. There is a very famous hypothesis proposed. Nobody really took note of that. That hypothesis was gene for gene hypothesis, right? Very few people paid attention to that. It's all done by a passionate worker, H.H. Floor, right? A series of 4 or 5 papers, all single author papers, and he came out with this theory. Now we realized whatever he hypothesized in 1945, everything is true literally. There is no mistakes whatever he proposed. Let's see what is that. What he told at the time, there is no concept of gene in those days. He told something like host conditioning factors. For resistance, he called a terminology, what is called as host conditioning factors. And pathogen side, he called again the pathogen conditioning factors. The host conditioning factors, hypothesized by Floor, is nothing but the receptors, or functional or proteins, or functional or gene, whatever, right? Or product of host or genes. The corresponding pathogen conditioning factors, beautifully he told, there is a corresponding pathogen conditioning factor. That means these two are matching. The host conditioning factor and the corresponding pathogen conditioning factor, that is nothing but the product of the so-called pathogen genes. They are effectors, right? So there is a receptor on one side in the host. We have effector on the other side in the pathogen. When they interact, the interaction always leads to no disease condition. So people thought there is something like a virulence factor which is operating in the pathogen. They called it as AVR genes because there is no proper explanation in those days. Now we know much better. The AVR genes and virulence genes, they are one and the same. There is no difference. It is situational. If there is a receptor, then it is a AVR is ensured, or no disease is ensured. If there is no receptor, then the susceptibility is ensured, right? So it is virulence or avirulence is not decided by the pathogen, but rather it is decided by the presence or absence of receptors on the host side. Try to take this point, it's the most important point, right? And when the host or gene product and pathogen AVR gene product participate in protein-protein interaction, and the outcome is accelerated defense all the time, provided the R protein is having the right configuration to come to a lock and key conformation with the effector protein. That's more important. So here comes the pathogen play the tricks. They modify the effector and come out with a different protein. So R protein becomes functionless. Then we call it as the resistance is broken down. It's a simple mutation event which is triggered in the AVR genes. It comes out with a completely different protein which cannot pair with the so-called receptor protein. Remember, R protein is still there, but it becomes non-functional, right? The mutation is taken place in the pathogen side. So the outcome is accelerated defense or the host resistance via cascade of signaling events, which I already told you, different series of events. There are a number of other genes also participating in that particular event, right? And several other plant genes also participating in the whole event. It's not only the R protein and the effector protein, they interact. Of course, they start the reaction, but it is guided by so many other genes from the plant side, okay? This is in a nutshell how plant and pathogen really interact and what are all the factors they guide that interaction. If you have any doubt, please clarify before I move on to the next slide. Anybody is having any clarification, please?
What is the difference between the PAM and the effector?
Okay, great. It's a nice question. I already told PAMs are extremely conserved on the microbial side. It is a general molecule which is present in most of the microorganisms, right? Effectors are something which is a specialty proteins produced by few elite microorganisms. Those elite microorganisms are called pathogens. You got my point? So we have more than 99.9% of the microbes cannot gain access to the plant sphere to take the plant metabolism for ride. No, because we have PTA, that's good enough. But the few, the minuscule of microorganisms, they acquire the capability to produce effector proteins. Only those microbes, they can negotiate with the host metabolism because the effectors are so powerful, they are delivered right in the plasm and cytoplasm, and they can manipulate the plant genes, right? They have access right into cytoplasm, and they have access right into nucleus also, because we know the talents. Transcriptional activated effectors are delivered in the nucleus. So you can imagine. So PAMs, they cannot enter into the plant system at all. Their interaction is only at the surface level, right? So that's a big difference, right? PAMs are general, effectors are very, very specific. An effector produced by one pathogen it cannot be recognized by a R gene of another host or R gene of a closely related host also. Not possible, right? They are very specific. The two major differences, one is highly conserved, another one is highly polymorphic, right? Effectors coding genes are highly polymorphic. They are vulnerable for any mutations, right? There is a point mutation in effector coding genes leads to emergence of new race. Is it clear to all of you? This is a concept I wanted to communicate to you, right? So let's move on. In the absence of functional R protein, I mean it here, there is nothing like no R genes. R genes are always there, right? The question is whether it is functional or not. So what is the meaning of functional here? Functional means a protein which is capable of interacting with the effector protein. That is what is called as functional, right? There is a correct protein conformation on either side. They can physically interact with each other. I'm not taking you to a tour of different hypotheses now proposed to explain how this effector and receptors are really interacting. So it's beyond the scope of this class. But you take the message, there is a kind of physical interaction which is taking place between receptor and effector. That interaction leads to a phenomenon what is called as ETI, right? So, but in the absence of a functional R protein, what I mean, a functional R protein is so important here. The pathogen-coded protein, also called as effectors, can trigger host susceptibility, right? Which I already told, via many other host factors. Okay? The cellular hubs I was talking about, the plant genes or plant gene associated factors, they are all going to interact with the effectors, or effector has got direct access to those domains. And when they have direct access to those domains, the outcome is always susceptibility. Who is going to block it? A functional R protein. If there is no functional R protein, the effector will have direct access to those domains which is going to contribute for pathogen life cycle completion. Is it clear to all of you? Let's move on to the next one. Now come back to the genome editing now, right? Now the story is out here, how the pathogen really interact with the host, who are all the main players here, right? PAMs and PRRs on one side, we have effectors and receptors on other side, and the third one is so many other associated plant genes, they participate in plant signaling pathways. So right now we have 3 plant associated genes or gene products to deal with. One is surface receptors, what is called as PRRs. Another one is specific receptors. They are called R proteins or intracellular receptors. And third one is a group of plant genes. They do ancillary role here in the signaling pathways. There are so many genes are there, they participate in signaling pathway. I was talking about ROS production. How ROS is produced? There is a handiwork of some genes there, right? There is a char induction, right? There is a lignification, cell wall reinforcement, callose accumulation, right? So many, phenol accumulation, phytolexin biosynthesis, right? They're all somewhere, some genes are expressing something that is playing a role there. And this group of genes are called signaling pathway associated genes. So now the choice is, you have 3 different choices at your disposal. One is surface receptors, another one is intracellular receptors or cytoplasmic receptors, and third one is a group of genes, they have an ancillary role, right? Let's see. For genome editing, for disease resistance, the choice of gene or genomic loci may be any one of these plant receptors. They participate in either PTI or ETI, right? So, how many different types? And any of the effector targets playing a role in ETS. I was talking about, in the absence of functional or proteins, the effectors will have direct access to certain cellular hubs or plant gene associated factors, right? If the plant gene, which is well defined and the function is well characterized, having a direct involvement in ETS, and all those genes can be targeted here, right? And plant cellular proteins or plant genomic regions coding for susceptibility factor, right? And this susceptibility factor is now called as S genes. S gene means the genes, plant genes, which is assisting the pathogen to take nourishment from the plant. It's very interesting. This plant genes are also doing the job against the plant, right? Those genes are called susceptibility genes, right? So, third one is any one of the different signaling susceptibility, different signaling or susceptibility signaling pathway associated factors, and modification of different signaling pathways. These are all the choices. Let's see what will happen if you try to edit or if you try to meddle with those receptors. We have to see the pros and cons of doing that. That's very, very important here, right? This is clear to all of you. Either PTI or ATI associated receptors can be a potential target, no doubt. And it can be those cellular hubs or proteins or the genomic region of the plant, they somehow assist the pathogen to undergo the transition from one phase to another phase. In the process, the pathogen is colonizing there, or establishing the symptom there, right? Those genes, right? They are popularly called as S genes, right? We'll come to that discussion. So, third one is any one of these signaling pathway associated genes, right? These are the potential target. Let us not forget, we are trying to manipulate the plant genes, right? And in my opinion, let us not meddle with the pathogen genes at all, because you cannot control them, right? You may control the isolate what you have, but what about the rest of the isolate? They are out enjoying the environment outside. You can't do that. It's very difficult. So what is manageable here is the plant genome that is right in your disposal, right? I hope I answered your question. So the editing of genes, which is coding for plant receptors. This is a kind of, in my opinion, when I was reading this, my mind was going to different directions. The direction one, is it really feasible, number one? If you do that, what are all the consequences? Let us have some discussion here. This I have borrowed from one famous paper appeared in Current Opinion in Plant Biology. If I'm wrong, please search this. The author is Barthoma from Wageningen. So he proposed the receptors, in particular RLPs and RLKs, are the potential targets for bringing a broad resistance in several hosts. Let's take that example now. Let's see, editing of receptors, right? So how many types of receptors are there? We have what is called as general receptors. They are on the cytoplasmic membrane. They negotiate with the PAMs, pathogen-associated molecular patterns. And we have intracellular receptors. There are two kinds I told, CC-NBS-LRR and TAR-NBS-LRR. They negotiate with a powerful pathogen molecule, what is called as effectors, right? They are specific receptors. Remember, general receptors are highly conserved. Mutations are not tolerated, please. You must understand, conservation means mutations are not tolerated. If there is a mutation, there would be a fitness cost. Come to the specific receptors, they are extremely polymorphic, much like the polymorphism what you see in R proteins, R genes. We know that R genes are highly polymorphic, right? And much similar to that, on pathogen side also, the effectors are highly polymorphic. Let's forget about effector now. We'll come to the receptor here. They are very specific, right? And base modifications or genome editing may be performed in pathogen recognition sites. What I want to make a mention here, in those receptors, please remember I was talking about ELRR in the case of plants, in the case of PRRs, and LRR in the case of R proteins. Remember, they are the place where the pathogen associated molecules are going to interact, what is called as pathogen recognition sites, PRS, right, in receptors, to enhance pathogen perception so as to trigger innate immune response. This is one hypothesis. You can bring a specific basis in those LRR so that you can increase the perception capabilities of those receptors. It's very interesting and revolutionary thinking, right? So what Barthoma really highlighted here was you can bring a specific base modifications. That's the reality in genome editing. So that would, in the process, it would enhance the perception. Remember, enhancing the perception would always lead to immunity. It may be a PTI or it may be ATI, right? So if perception is not happening, it might lead to susceptibility. I already told, if there is a change in the pathogen effector protein, what do you mean by that? The perception is lost. The R protein is unable to see the effector because the protein is different protein. It comes with a different pattern. So it is unable to perceive what is coming inside. That ensures ETS, right? So what this paper says, you change the perception capabilities of those effectors. Not only that, those R proteins, non-functional R proteins, you can bring life to the non-functional R proteins just by changing the bases there. It's really a revolutionary thinking. In my opinion, it is quite a reality. That's number one. So this approach is being explored to mount multiple pathogen perception. That's also wonderful thinking. Because most of the R genes cloned and sequenced right now, they have the same backbone, NBS, LRR, right? If that is the case, definitely you can bring subtle changes in the perception PRS that would bring a capability to this receptor to recognize many pathogens. So that is also another option, right? But in my opinion, this needs to be done with a great care. We know pretty well if a small amino acid change in the effector makes the R protein non-functional. If you do the same thing in the receptor side, the same thing can happen there as well. Right? This needs to be done very carefully, as receptor modification must not lead to effector non-perception by edited receptors. Try to understand this statement, right? If you edit the receptor, in the process, whatever protein is going to come out of this receptor should be able to retain the perception capability of the effectors, right? This is one of the most important points. Otherwise, the ETS would be ensured here. Whatever effort you are doing is going to be a counterproductive operation, right? And therefore, the correct identification and modification of PRS assumes crucial here for any success, right? This is one option, right? And this is the broad options available for us right now. Receptor or resistant gene editing. And what is suggested here is what is called as a synthetic resistant plant, where you try to modify the receptors to bring more and more PRS into the same receptor. In the process, the plant becomes resistant to diverse group of pathogens, right? That's what it is here. Genome editing for single resistance and genome editing for multiple resistance. If you deal with the PRS, you just modify the non-functional R gene, base modification that leads to a functional R protein, then that is going to trigger ETI in the host. So when the ET is the most desired trait, because we need that more and more in the crop systems so that the pathogen can be controlled very easily, right? But in the case of multiple, what he was suggesting is a potential use of editing technology is engineering a novel synthetic R proteins, R genes able to mount resistant to several pathogens by combining PRS from different R genes, right? Here comes the resources what is available at our disposal. Here, most of the genomes are already out, and we know what is there, right? And getting a kind of a diverse R protein from a particular host is quite a reality now, right? So if you know the PRS and you can do the modification in one R protein, R gene, you try to bring modification in the PRS, which is going to perceive a diverse pathogens. So that means what? A single R protein would do multiple pathogen perception, and it would evoke a similar stereotypic response in the form of ETI. So that is what it is needed here, right? And this is a paper, Current Opinion in Plant Biology. If you have time, it's a very easy to read, nicely written, and he gives a complete account of receptor modifications and receptor editing of receptor coding genes as a solution for mitigating more than one pathogen in a crop plants, right? So I'll move on to next important target. Perhaps a lot of work is done now, what is called as editing of the genes which is coding for susceptibility factors. The success whatever we have in the form of product, in the form of success stories are all from this particular manipulation, right? S gene manipulation. So editing of effector targets or S genes using modern omic platforms, several susceptibility genes have been identified now, providing many potential targets for improving crop protection. And if you see the last 5 years of papers, you see more and more of genome edited crops, in particular with the help of CRISPR-Cas9, and the products are already out. I'll show a couple of them here. The unprecedented efficiency of genome editing techniques in editing the specific sequence of S genes, which represent the best candidate for engineering resistance right now, right? What's happening here is you edit those plant genes, which is going to interact with effectors in the absence of R proteins. And if you edit them and make the factor which is not palatable for the pathogen, it's a very simple concept, right? So it has conferred resistance in various crops. I think a number of references are there. So these are all some of the success stories which is already there, and different crops. I'm not going to read it here. I'll come to some specific cases here. And these are all the success stories in different crops, like you have tomatoes, grapes, apple, wheat, and rice and tomatoes and citrus, a lot of success stories, right? And a success story with the S gene or promoter sequence editing. Let's take 3 examples here. The effector target the plant cellular hubs, just to rewind what's really happening. And these susceptibility factors are the potential targets. And the classical one, which is already there in a number of applications, is MLO. Right? I think you please put this as a search word that will take you to a world of MLO loci in different crop plants, right? So MLO locus is the best studied one, and there are 7 clades, and clade 4 and clade 5 are so important because this clade 4 group of S genes, they assist a powdery mildew pathogen to cause disease in the plants, especially in the case of monocots. Remember, powdery mildew of wheat is #1 problem in many places, right? Powdery mildew of wheat, Bloomeria graminis, forma specialis triticine, right? And the clade 5 is for dicots. We have powdery mildew in number of dicot plants also, right? So this is a clade. Somehow the clade 4 and 5, it evolved in those plants, excuse me, to assist the powdery mildew pathogen to cause disease. And they are bona fide susceptibility factors in those monocots and dicots, right? And people have attempted to manipulate this MLO loci, and the phylogenetic analysis of 341 MLO proteins from different host plants to uncover the evolutionary history and origin of MLO family. During evolution, the family has diversified into 7 phylogenetic clades, with the clade 4 and 5 are so important for us because they are directly involved in assisting the powdery mildew pathogen to cause disease in very important, agriculturally important crops like wheat, and also so many diverse crops in dicots as well, right? And they help the powdery mildew pathogen to infect. And this is a typical, what do you call, MLO. The gene was mapped into the genome of higher plants. MLO encodes a plasma membrane associated protein. that binds a calmodulin with its C-terminal cytoplasmic tail. Here is a calmodulin binding, a domain that is relatively well conserved in the many of MLO family. And this protein is structurally related to G protein coupled receptor in metazoans and the function in susceptibility towards powder mildew in number of plants, right? So this is a typical MLO characterized one. So some information about this MLO. This is mildew resistant locus O, and one of the best known as genes, and it's a prominent example of robustness in durable pathogen resistance program. The wild type alleles of MLO was first discovered in barley to confer susceptibility in barley against powdery mildew, and that is Plumeria graminis, Forma specialis hodii. MLO encodes a membrane associated protein with 7 transmembrane domains. So you can count there are 7 here. So it is conserved throughout monocots and dicots, and it is required for mildew fungal penetration under the host epidermal cell. I was talking about there is some function they confer to the pathogen. Here it is penetration of host epidermal cells. The mutation in MLO with the CRISPR-Cas9 has also conferred mildew resistance in wheat and tomato in a non-transgenic systems. So this is in a nutshell. I don't want to take the too much of time on this. You know it very well. They have edited this MLO loci, and they have confirmed it through a number of approaches. And they have sequenced it, and they found that around 48 to 49 base pair deletion, chromosomal deletion in this MLO loci, that gave a complete resistance to powder mildew. That's what it is highlighted here, and they named this particular variety as a tomello. The tomello is a variety which is an edited tomato line which gives a complete resistance to powdery mildews. Okay, they have edited the MLO loci, right? The leaves of edited tomato line infected with Oidium neolycopersicea, showing the full resistance towards this pathogen when compared to wild type leaves, right? So next example is enhanced disease resistance, or also called as the EDR1, that is in wheat, again for powder mildew. A raft-like mitogen activated protein. They have been targeted by CRISPR-Cas9 and significant reduction in mildew in wheat. The thumping success, what is reported in the recent times by several groups, is in the case of rice. So I'll take you to a few slides on this success story in rice. In rice, there is a group of plant genes. They help the pathogen to sustain this ETS. In the process, the symptom appears, and those genes are called sweet genes, right? The pathogen Xanthomonas oryzae, Pathovar oryzae, it secretes one or more 6 known transcription activator effectors, what is called as a tails. This tails has got a very specific target in the host genome. Those targets are called sweet genes. Remember, sweet genes are host associated genes, and those genes products are specifically targeted by these effectors. The effectors are transcription activator like effectors, and these effectors, they bind to the specific promoter sequences and induce the disease blight in the rice leaf. There are 3 genes are well documented: sweet 11, 13, and 14, and people have touched those genes 13, 11, 13, and 14. CRISPR-Cas9 mediated genome editing has been done to induce mutations in all 3 sweet gene promoters. Remember, they have touched the promoter region, and editing was further confirmed by sequence analysis of they have confirmed it, and the trials they have conducted, the edited line, and they found that there is a broad spectrum resistance against diverse Xanthomonas isolates. So that is, I think they took around 63 in place, and they found that the edited lines are resistant to 63 different Xanthomonas. That is something we need to look into, because the ATA-based resistance program is extremely vulnerable. I was talking about a single point mutation would make that ATA functionless. But in this case, just imagine a small mutation in the host side, a sweet gene is conferring a broad resistance against diverse isolates of the same pathogen. That's the power, because I was talking about bacterial pathogens are so diverse and they are variable all the time. So this approach, perhaps one of the potential approach for bringing a broad resistance in a crop like rice. This is in a nutshell the regions, and the effector in question here is tails, and the effector target is sweet genes, and the interaction which leads to ETS, and the genome editing option as prevented the ETS from happening in the host. That's what they did in a nutshell, right? They edited the sweet genes. In the process, what happens? The interaction is broken. There is no interaction between the tails and also the sweet gene products. So the ETS is stopped abruptly. So the mutation in sweet gene makes the plant completely resistant. That's what the story. So the genome edited zoo lines, they didn't stop it there. They took it to the mega varieties like IR64 and Chiharang sub 1. And the same thing they did here, and they come out with the so-called completely resistance against bacterial blight in rice. Right? And they also did a lot of other agronomic traits, yield potential, quality, and everything seems to be same as the kind of wild type 1. Here I must make one important point here. This is a slide what I borrowed from that particular paper, and you can see here there's a complete resistance on those edited sweet gene edited lines in rice and against diverse Xanthomonas isolates, right? So we'll move on to the third approach. Not too many papers are there in the literature tackling or editing the genes which is associated in the signaling pathway. I just brought a couple of them. NPR1 is a kind of a gene which is participating in number of signaling events in the plant. You can think of something like this, and the TGA transcription factor, we know it very well. They participate in SAR and salicylic acid mediated resistance. And VIRK70 is another transcription factor, can be tackled. These are all some options, and I could not find a kind of thumping success like the one what we found in S genes. So I'll just stop it here. And these are all some of the success stories in agriculture, in particular fungal and bacterial disease management using genome editing route. So the list is there. Don't try to read it here. All papers are in public domain. You can read at any time. And I just would like to make a point here. This particular table, what is dominating here is the CRISPR-Cas. So that is the power, right? So although the talents are here, 2 talents are here, but most of the success stories are coming from CRISPR-Cas. That is the point. And the loci, what they tackled here is also here. And again, if you see, all of them are associated with the susceptibility factors, right? So the product what we have or what people witnessed in the form of success stories, most of them are from the so-called ETS-associated growth plant factors, and popularly called as susceptibility genes, right? Whether it is MLO1, MLO1 from Triticum aestivum, from Solanum lycopersicea, and EDR1 from wheat again, and there is another loci here, ACRF in the case of rice. So they have given a broad spectrum resistance against all those paths. This is another again the list what is being reported. And another interesting story is most of the reports what we are witnessing now is coming from 2016 onwards. So that means there's a great deal of work is really going on in the last 3-4 years to come out with a product, right? So that is all about the success and promise what we have witnessed using the revolutionary genome editing technology. But it comes with a lot of the flip side also. It is all is not well all the time, right? So if you're lucky, yes, your agronomic traits are really good. And in addition to the edited line, that is the most desired one. You try to edit for pathogen resistance. Okay, pathogen resistance is done. But in the process, if the plant is not growing, then it is of no use, right? So one of the most important highlight of this genome editing, in particular with the help of S gene, is the fitness cost. Let us not forget, there may be a multifunctional S genes. They have more than one function. In addition to guiding the pathogen, they may have some vital role in the very developmental process of the plant itself. If you touch them, yes, you get a resistant line, but in the process, the plant may not look like the plant again. It may be maybe something else, some handicap, maybe some fitness cost. So the choice of S gene becomes so important, right? So there comes the role of individuals who is touching this kind of work. They need to do a thorough literature search. They need to go for maybe a different isoform which is not having multifunction, and they need to touch the one which is having exclusive role in the so-called virulence or the susceptibility. Then the success is ensured, right? So this is a kind of a slide, the comparison. I think you can make your own points here. It's not my points here. Of course, the traditional one is very slow. Big efforts are needed in terms of money and time, and only R genes are addressed here. The classical breeding program, we address only the R genes, and the undesired traits and phenotypes need to be removed through different kind of backcross methods, and that delay the arrival of your variety. And in my opinion, they are not really durable. If you put the time scale of a decade, they are not durable. They may work for maybe a couple of years or so, but if you put a decade, it may not work, right? And in the classical, I always tell this point, SR32, 31, in the case of wheat, it was resistant for 30 years. On fine morning, UG99 came and make that particular resistant gene non-functional, right? That means it is always in the time scale. Any cultivar may become redundant or become, not so useful in agriculture. So in the case of S genes, perhaps that promise, such an approach, such a lacuna may not be there, because the paper says that sweet gene editing gives resistance to 60 plus isolates of xanthomonas is a very strong message, because the durability is already there, right? So that is an important point. So edited lines, editing option is a rapid, quick, no restrictions either on R genes or S genes. Choice is yours, and precisely controlled, less off or non-target, because you are specifically modifying that particular gene. The precision, and also no off-target is ensured here. Durability is also there in the case of S gene-based one. So I think that's the last one. I think the summary is written here. Definitely this CRISPR-Cas9 system become a gold standard method for genome editing for various reasons. I think different speakers might have highlighted this in the same platform. And the simple, efficient, and versatility. And genome editing has been applied to enhance plant resistance against a number of pathogens. I showed several slides here. And susceptibility genes are the prime focus right now, provided they are having a very narrow spectrum of activity only to guide the pathogen to cause disease. They do not have any other multifunction. Those multifunctions should not be related to the plant developmental process. And the current legal framework, of course, maybe yesterday there was a talk. I think you might have listened to the talk. And definitely in future, the bio-edited disease-resistant crops will become a standard tool in plant breeding programs. I think the effort by NHP cast in IRI is a small step to instigate the young minds to initiate such a program in the years to come. So with this, I thank you all of you for, it's a pretty extended lecture. I do not know whether it really benefited all of you. So with that, I close my lecture. I thank organizers, in particular Dr. Vishwanathan, Ramcharan Bhattacharya, and Anirban for giving me a platform to share my thought process. And if you have any quick questions, I'm ready to answer here, please.
Thank you, sir, for your lecture. It was good for those who do not know in this area how the things are working. So the mechanism was very nice. I exactly want to know how these effector molecules are produced. So means after infection they are produced, or how they are produced?
Yeah, good. It's a nice question. For effector genes in the pathogen, they are not really needed for the pathogen in the true sense. There are so many other microbes, they live happily outside the plant system. Right? They do not have effectors, or rather functional effectors are not there. Still they lead a happy life. But why few microbes evolved effectors? That is in the evolution. They evolved these gene clusters only to take advantage of the host for their perpetuation, number one, their multiplication, colonization, and the very disease. What is after all disease? Enlarging its population so that the progenies are sustained in the environment for a long time. Effectors are typically expressed when the pathogen is in the interior of the plant system, right? Remember, effector genes are in the pathogen. Their expression is always happens when they interact with the plant endogenously or wherever the pathogen is adapted, maybe powder mildews in the epidermal. So when they interact with the pathogen, there's a physical interaction. Maybe the PTI signal is somewhat linked to the effector gene expression, right? There must be a trigger for the effector gene to express. Remember, number one, once it is expressed, there are endoplasmic reticulum in the pathogen. By and large, they are produced there, and it is transported outside the pathogen system into the plant body. In the bacterial system, it is thoroughly worked out. Most of the plant pathogenic bacteria they have a syringe kind of stuff on their surface, what is called as type 3 secretion system. The type 3 secretion system, if you see the electron micrographic photo, it is typically like a syringe, right? This is exploited. So the type 3 secretion system and effector secretion is all coordinatedly expressed. When the effector is released, biosynthesized in the pathogen, upon some trigger which is coming from the host, perhaps it is from PTI, we are not very sure, the effector is ready, because effector is not needed for the pathogen at all, right? It needs to be transported outside the pathogen into the plant. That is mediated with the help of type 3 secretion system in the case of bacteria. In the case of fungi, all of you must be knowing the fungi produce a specialty structure what is called as hostoria. This hostoria is a kind of invagination in the cytoplasmic side of the plant cell. Just imagine the textbooks. There is a hostoria which is coming into the plant's cytoplasm. And in that, the hostorial boundaries, that is a transaction point between the pathogen and the plant, right? The effectors are released into the plant side through hostoria. The mechanism is still to be worked out, but that is the transaction point between the pathogen and the plant cell. The hostoria is the one which delivers the effector into the plant cytoplasm. In the case of nematodes, we know that all stylet-borne nematodes are plant pathogenic one. It is something like a syringe that stylets are the one which releases the effectors into the plant system, right? This is how how the effector is delivered. In the case of fungal disease, viral diseases, you know it. There is an insect which is injecting the virus right into the cytoplasm. Yes, if at all any virus coding effectors, they are directly delivered into the cytoplasm, right? So there are too many ways of delivering the effectors. The pathogen evolved in their lifetime. They know how to negotiate with the plant cytoplasm and the nucleus also to take the advantage for them, right? Yeah.
Is it possible to block the release of the effector?
Yeah, the so-called origin-based one is that only. So what happens is it is not blocking the effector biosynthesis. Rather, we need more effectors actually to trigger ETI, right? We need effectors. Without effector, ETI will not be triggered, right? And there is no point in manipulating the effector coding genes. It's not needed actually, right? Let the effector be there, because I already told in a PRS we need more and more perception by the R genes. We need effectors, because when they interact, then only the ETA is going to be triggered in the plant system. So there need to be an effector. I was talking about ETA and ETS, but for effectors, there is no ETI, there is no ETS. We need effectors, definitely, no doubt. And equally what we need is the receptors, which is able to interact with the effector. In the process, the ETA is triggered in the plant system, right? Yes, please.
Sir, very nice and very informative lecture. As mentioned, Dr. IARI, you are an excellent teacher. It's reflected here. So, sir, I have like one question. In case like plant immunity, so as you mentioned that it will, if ROS production is there and if plant can fight that. In some cases, if you see if you working in, if I have working in diabetic stress field, so if we overexpress some of the gene that can fight with ROS and all that things, and minimize the generation of ROS, like although some amount of ROS need for signaling all that. So my question is that if you working and overexpressing any of this abiotic stress gene, and in that case, if you can minimize the ROS production in plant, so how can we check whether this gene having like also resistance to some of this biotic stress also? So in that case, if you...
Yeah, I'll sum it up. I think you want to reduce ROS production in the plant system so that the biotic stress, let us not forget, RO is a defense signaling, a defense molecule. It is needed by the plant.
At a small amount, it will be needed.
It is needed by the plant, definitely. So otherwise, the ROS is not a component of this defense signaling pathway, because they are very powerful antioxidants, I would say antimicrobials. They play a dual role. First, they themselves are antibiotic, number one, and number two is they can participate in signaling events. They do both the functions. But in your question, I don't think I will be able to answer that. How to manipulate or regulate the ROS production in the plant system, I don't think I am competent to answer this question. But yes, if anybody who has some knowledge, you can interfere here.
There are several crosstalks between the abiotic stress signaling pathway signals and the biotic pathway signals. There are several such kind of molecules, particularly different kind of this hormone signaling receptors and other things. They are very much common between abiotic and biotic stress. Definitely, if you do some modification, one of such pathway related genes, you can have resistance against both the